wolf's egg, we may give birth to an animal that hasn't walked the earth since 1936. And then you can start going back further, and you can start thinking about dodos. And you can think about other species. And in other places, like Maryland, they're trying to figure out what the primordial ancestor is. Because each of us contains our entire gene code of where we've been for the past billion years, because we've evolved from that stuff, you can take that tree of life and collapse it back, and in the measure that you learn how to reprogram, maybe we'll give birth to something that is very close to the first primordial ooze. And it's all coming out of things that look like this. These are companies that didn't exist 5 years ago. Huge gene sequencing facilities, size of football fields. Some are public, some are private. Takes about $5 billion to sequence a human being the first time. Takes about $3 million the second time. We will have a $1,000 genome within the next 5 to 8 years. That means each of you will contain, on a CD, your entire gene code. And it will be really boring. It will read like this. The really neat thing about this stuff is that's life. And Lori's going to talk about this one a little bit. Because if you happen to find this one inside your body, you're in big trouble, because that's the source code for Ebola. That's one of the deadliest diseases known to humans. But plants work the same way, and insects work the same way, and this apple works the same way. This apple is the same thing as this floppy disk. Because this thing codes ones and zeros, and this thing codes ATCGs, and it sits up there absorbing energy on a tree. And one fine day, it has enough energy to say, execute, and it goes, right? And when it does that, pushes .exe, what it does is it executes the first line of code, which reads just like that, AATCGGACCC, and that means make a root. Next line of code, make a stem. Next line of code, TACGGG, make a flower that's white, that blooms in the spring, that smells like this. In the measure that you have the code, in the measure that you can read it, and by the way, the first plant was read 2 years ago. First human was read 2 years ago. First insect was read 2 years ago. First thing we ever read was in 1995, a little bacteria called Haemophilus influenzae. In the measure that you have the source code, as all of you know, you can change the source code. And you can reprogram life forms so that this little thingy becomes a vaccine. Or this little thingy starts producing biomaterials, which is why DuPont is now growing a form of polyester that feels like silk in corn. This changes all rules. This is life. But we're reprogramming. This is what you look like. This is one of your chromosomes. And what you can do now is you can outlay exactly what your chromosome is and what the gene code on that chromosome is right here and what those genes code for and what animals they code against, and then you can tie it to the literature. And in the measure that you can do that, you can go home today and get on the internet and access the world's biggest public library, which is a library of life. And you can do some pretty strange things, because in the same way as you can reprogram this apple, if you go to Cliff Tabin's lab at the Harvard Medical School, he's reprogramming chicken embryos to grow more wings. Why would Cliff be doing that? He doesn't have a restaurant. The reason why he's reprogramming that animal to have more wings is because when you used to play with lizards as a little child, and you picked up the lizard, sometimes the tail fell off. But it regrew. Not so in human beings. You cut off an arm, you cut off a leg, it doesn't regrow. But because each of your cells contains your entire gene code, each cell can be reprogrammed if we don't stop stem cell research, and if we don't stop genomic research, to express different body functions. And in the measure that we learn how chickens grow wings and what the program is for those cells to differentiate, one of the things we're going to be able to do is to stop undifferentiated cells, which you know as cancer. And one of the things we're going to learn how to do is how to reprogram cells, like stem cells, in such a way that they express bone, stomach, skin, pancreas. And you are likely to be wandering around, and your children, on regrown body parts in a reasonable period of time. In some places in the world where they don't stop the research. How does this stuff work? If each of you differs from the person next to you by 1 in 1,000, but only 3% codes, which means it's only 1 in 1,000 times 3%, very small differences in expression and punctuation can make a significant difference. Take a simple declarative sentence. Right? That's perfectly clear. So, men read that sentence, and they look at that sentence, and they read this. Okay? Now women look at that sentence and they say, wrong. This is the way it should be seen. That's what your genes are doing. That's why you differ from this person over here by 1 in 1,000. Right? But, he's reasonably good looking, but I won't go into that. You can do this stuff even without changing the punctuation. You can look at this, right? And they look at the world a little differently. They look at the same world and they say, that's how the same gene code, that's why you have 30,000 genes, mice have 30,000 genes, husbands have 30,000 genes. Mice and men are the same. Wives know that. But anyway, you can make very small changes in gene code and get really different outcomes, even with the same string of letters. That's what your genes are doing every day. That's why sometimes a person's genes don't have to change a lot to get cancer. These little chippies. These things are the size of a credit card. They will test any one of you for 60,000 genetic conditions. That brings up questions of privacy and insurability and all kinds of stuff, but it also allows us to start going after diseases. Because if you run a person who has leukemia through something like this, it turns out that 3 diseases with completely similar clinical syndromes are completely different diseases because in ALL leukemia, that set of genes over there overexpresses. In MLL, it's the middle set of genes, and in AML, it's the bottom set of genes. And if one of those particular things is expressing in your body, then you take Gleevec and you're cured. If it is not expressing in your body, if you don't have one of those types, a particular one of those types, don't take Gleevec. It won't do anything for you. Same thing with Herceptin if you've got breast cancer. Don't have an HER2 receptor, don't take Herceptin. Changes the nature of